So we're right now exploring Point Doom. We know it contains methane seeps, and we're doing geochemical and biological exploration of this region.
So
That's
that's
huge
a
map.
Wow.
Yeah.
Look at that.
Are these?
Even an Argus shot, it looks great. Look
Wow,
at that.
yeah.
Wow.
Oh, yeah.
It's like a snow patch. I'd say.
Yeah, this is
Wow.
great.
The mixture of orange
We
and
zoomed in and
white.
it looks like clam.
You want to zoom in again?
Oh, no, sorry.
Yeah,
Okay.
let's do it.
Yes and no.
Yeah.
We'll go with yes. Look
Always yes.
Jeff's always going to say yes.
Whoa.
at that.
That was really cool looking. Wow.
Yeah, so we can sort of see the clamshells there on the
Yeah,
right.
it's just like the tips of
Science
clams, I
Snap,
guess.
did you want
They
to hold
worms,
here?
maybe?
No, there are
'cause they're
snails
often
too.
buried sort of vertically into the sediment, so that
We're
their
going pretty
feet
slow,
won't
Emil.
So there's
get
like
the sulfide.
there's also a ton of snails around too, though. I think.
Yeah, this is
Right,
all right.
covering
Wow.
them.
Yeah.
It's a
Oh,
good
look at
smaller
that.
observational
feet there.
speed.
Yep.
It's great.
Yep.
Great.
You're right.
Look at Argus view too. That's
Yeah.
definitely a bacterial mat highlight.
Yeah, that's fantastic.
Good focus, Carrie.
Wow, looks
Oh,
like
it's quick.
snow.
Whoa.
Oh, no.
So sad.
We made
Who
brown.
did
I'm not
that?
even close.
It's ballet.
Bits of that.
Material mat ballet.
Yeah.
I'm
So-
turning Argus, by the way.
Yeah, it's very
Roger.
feathery. That's cool.
I'll move along.
Cool perspective.
Cool.
Oh, that's like kind of a strange.
Yeah, it's interesting.
Did you
perspective.
see that
It
weird-
Felt like it was a
Yeah.
Wait,
Optical illusion
what was
there.
the
Yeah,
illusion?
that was a
That's
weird
with me, yeah.
illusion.
What was happening? I still haven't figured it out.
It's those pieces that were floating up, I think.
Oh.
Distorted everything.
Look at the size of this mat.
Wow.
Wow.
This is
This is
great.
incredible.
I can see why we came back here.
This is dreamy.
This is wild. Yeah.
It's pretty great.
So, Jeff, the clams that live
like
down
a
here,
map.
and maybe some worms, are they feeding on the bacterial mat, or do they have symbiotic
Yeah.
bacteria in them? That
The clams, at least, mostly have symbiotic bacteria. So
Okay.
they're the, again, sulfur oxidizers that are
Don't go too fast.
able to,
Roger.
on the clam foot, kind of dig down into the sediment to get the sulfide. And then the animal as a whole is therefore able to access
What's our
the
bearing?
oxygen
Our
in the
bearing
water.
to-
So,
Oh, our heading?
yeah.
Yeah. Well,
Well, or the course is, we're heading southeast.
Okay. we doing a core or slurp or what's the scoop?
So yeah, we're going to want to get an EH measurement
Okay.
for about 5 to 10 minutes and then take the core. So
All right.
EH is a measure of the redox potential. So that's a sense of kind of how willing the environment is to give electrons or take electrons. And electron movement is really what drives all of life, essentially, by moving electrons from one molecule to another and ideally gaining the energy in between. That's how all cells can survive. So if it's kind of a high EH, that means that the environment is oxidized and wants to take electrons. If it's really low, it's the opposite. It has a bunch of extra electrons. And we'd expect a reduced environment like this with low oxygen and a bunch of sulfide to have a pretty low EH value.
All right, Jeff, are we going for a black area or the orange area?
The orange area. Yeah, so once we're hovering pretty close right above it, we can start the clock.
We're just
So-
going to get the RV lined up, and then we're going to... Oh, are you following?
We enter this as a sample, or?
This is the EH meter that
Right.
we just want to sort of have
Yeah.
measurement for about 5 or 10 continuous minutes.
Just wondering if you want a log of the Latin long where this was, yeah.
Yep, that'd be great. Because we're going to take a push core next. Yeah, so as beautiful as these microbial mats are on the surface, I would argue that the real magic is happening beneath the surface.
There's magic.
Where the
Microbial mass.
You're saying
not
the
happy
beauty is
with
deep
how
within.
far off we
That's
are.
right.
What's going on underneath the surface?
That is
Well, I
where
can keep going.
anaerobic methane oxidation
Yeah.
is happening.
I think we should go forward a little bit.
So
and then down a
are
little bit.
are these
Roger.
mats on the
Going
is
forward.
the surface actually not the
30
big
centimeters.
bacteria?
No, it's definitely bacteria, but the below is this tight symbiotic relationship
bring us up.
of anaerobic methanotrophs,
That's okay.
and those are
That's good.
archaea and
Oh.
sulfate
See how
reducing
that settles out.
bacteria.
Cool.
Roger.
And they're eating the methane and making the sulfide that then drives
Do you want me
these
to come down a
mats
bit? I
that we
see
see on
your
the surface.
shoulders pretty.
Yeah,
Jeff,
my
what
shoulders
make
all the way
archaea different than bacteria?
So back up a bit.
You're
Okay.
clear.
Yeah.
Sorry, this
down
is really
10, maybe 20 centimeters.
I that's cool. I thought that anaerobic
Get back to that.
had to be, down in this deep in the sediment. It's right at the surface under the.
Yeah,
Bacterial
usually,
mat.
I mean, you do have to get below the surface a bit, but probably within
Get
2 or
pulled
3
back down.
centimeters here.
Oh,
Can't win.
In opposite zones, yeah.
is it sensitive to the very
I realized
sensitive to the
we were-
distance?
Oh, that's a
Haven't
hatchet
looked that up
fish
yet.
swimming back there.
Where?
It's hard to say.
Oh,
yeah, I'm
cool.
not
Oh,
sure.
that's cool.
Good,
Butterfly
good
thing?
spot.
What are we looking at?
In the top right corner, there's a hatchet fish, but it's like perpendicular to the camera right now, so it's really, really thin.
Oh, yeah.
Those are
Use
some of my
bubble.
favorite fish.
Looks like a moth.
And it's totally
like a
eating
moth,
the
yeah.
seafloor.
That's
That's
cool.
crazy.
Oh, cool.
Whoa. Digging in.
All right, here comes the core.
Yeah.
Okay, let's
Rock the porch back in.
Uh-huh.
Or
Yeah,
camera
roger.
out.
Okay.
Yeah.
There's a jelly off in the distance. That's cool.
Okay,
Do
we
you guys
need
have
to
an opinion about to the right
come
on that
point down a
patch
bit so we can
that
figure
we
where we
can't
wanna
see right
go.
now?
All right. We lost our hatchet
So-
fish.
Well, I think
no.
Pete can hear you if
So
he's
you
listening.
thinking,
I think he's listening, but you can.
right here might, ooh, right where those lasers are now might work. Right there.
So
You
we
can zoom there.
Oh,
We can go there, we can go further to the right.
I can't. I don't have any perspective.
Is that going to be all right? Paging science.
Yeah, sorry. We're learning how to use buttons.
Learning how to use buttons.
So it's complicated.
Is that going to be too disturbed for you? Is that going to be all right there?
Can't hear you.
Sorry.
He said,
Very
"Can you
good
zoom
job.
out a little bit?" And you
And here we
did.
are.
There you go.
Yeah, let's turn off the data
We'll do a
lab
little
one.
of that.
Okay, get rid of
Oh,
that.
there's the hatchet fish.
Sorry.
Yeah,
Oh, the
Yeah.
hatchet fish is dead or something.
Oh, here we go.
I
That
don't
is
think there's
solid.
Dang.
very much-
Okay. Not happening here.
No.
Darn.
No.
Bummer.
Unless that's what you want. Which doesn't matter.
Might be.
Yeah, let's maybe
definitely
if we could fly
slurpable,
back
but it's
toward that huge
Pardon me?
snowfield we saw. All right, so we're still looking for kind of a nice orangey spot.
Anything calling out to you?
Let's keep. I mean, that bit right in front of us looks potentially good.
Okay.
So Jeff, do you think that these mats are toxic to animals?
Or humans?
Or to humans.
Or
Give me
to
artist
humans?
back.
a good question.
Humans, whatever. But I'm wondering about that hatchet fish.
Yeah, right. Or that jellyfish.
Or
that's
that
not looking so
jellyfish.
Oh, come on, Argus.
happy.
Cat shark.
There you go.
So sulfide is toxic to most organisms.
Okay.
So the sulfide itself would be a problem, but
Mhm.
I mean, presumably these bugs are eating the sulfide, turning it into something else.
What a glamour shot.
I
Yeah,
imagine
I think
there's
they tried
still
that.
some around, and that if you take a bite out of the mat, you'd definitely be getting some sulfide
Yeah.
too. So
Yeah,
it's probably not
Ah.
great.
I've never seen a seascape like this.
Yeah, that's wild.
it's incredible.
Yeah, maybe.
Oh, there's something underneath that. Oh no, it's just the
Yeah.
slight currents.
Toward us a bit, kind of just where it starts to get a little bit more densey orange.
Okay. Is that lighting or is that actually densely orange?
Oh.
Get in there.
So in case anyone was wondering what was happening here, with all the
all the way
yeah.
in.
Oh, baby.
All right.
Hey.
All right.
It's not
Awesome.
landed yet.
Oh,
Stay in there.
Wow.
oh, so black.
Wow, that's super.
Do you want to keep on
Stay
Argus?
in there, stay in
Wow,
there.
is that
yeah,
ever
for now.
dark?
Is that a black
For now.
smoker?
Let me
Yeah,
know
I've
when you want
never
to
actually
change.
seen the dark black layer go all the way down.
Ah.
Usually it's a few centimeters and then it
Ah.
gets gray.
Uh-huh. Cool.
Can't.
That's a really nice shot of the sort of fibers
Yeah.
Yeah.
in the mat.
Oh,
And those are
shimmery
those
water?
kind
Wow.
Or
of
is it just jiggly?
sheaths that allow the microbes to transition between the sulfide-rich layer and the more oxidized surface. So they,
Yeah, that's really jiggly
It's jiggly,
sediment
yeah.
too. That's funny.
yeah,
Just from this. Oh, watch you're not halted.
That's,
That's
really
exciting.
cool.
that is really nice.
How
Okay.
close would you like to get?
Is that
That
good there?
looks pretty great. Yep. I'm impressed you didn't disturb it.
So do you get to lock it now that you're not floating around?
Absolutely.
Hey.
All right.
Right, yeah. Now we wait.
10
At
minute
exactly 7 p.m.
break. Go get yourself some cake.
It's about an inch off the seafloor. We can go closer if you'd like.
Do we see this thing crawling around?
Yeah, let's zoom in on that.
Is it possible to zoom in on that?
What are
while
we
we're
looking at?
holding
Just to
this?
the right of.
Oh yeah, absolutely. Let's get in on
It
that.
looks like
Creepy
a.
crawly.
Yeah, it's yucky.
There's also like these
It's
tube
yucky,
worms
it's
up there.
cool.
What are those guys up there?
Like
Oh,
some
yeah.
bacteria.
Yeah, those are some
Oh
tube
yeah.
worms.
Is that a polychaete? Is that? What? Yeah, it's got.
Yeah.
Not a crustacean.
So what's on the top left corner there? That little tube wormy
Yeah,
thing.
we're just looking at that.
Oh, that's interesting.
A tube worm thing.
I think it.
think
Oh, I
that
was
full
about to
zoom
say a
there.
siphon from a clam, but they.
They look big
not-
compared
yeah.
to the
I do
size of
see
the
some
clams.
clams in there.
I think it could be a.
And even the ones right above look like smaller versions of these, but that looks like a clam.
Yeah.
I don't know.
Is that our dead
But
hatchet
it's still
fish?
a
It's still
little alive.
ventilating, so it's still alive.
What
Yeah.
kind of fish is that? That's
It's a
what I saw
hatchet
before.
fish.
Hatchet.
Almost looks like a
Um,
candy wrapper.
Sternoptychidae is the family.
Huh.
Maybe.
Huh.
These are quite common mesopelagic fish. You can
If I can come back a bit.
Yeah, it
kind
does
of
not
see
look
along
happy.
its belly that it has little
That's all you got.
photophores. Right along the belly
left.
has little light-producing organs.
Oh,
We've
cool.
done 10 minutes also, by the way, science.
Yeah, so if we can zoom out and sort of measure how far we are from the hole, that would be great. Looks like it's about 15 or 20 centimeters, but we should record that if we could.
Yeah, I'll get the
All right.
lasers on it there.
Lasers.
20.
There you go. You want to screen grab that maybe, or zoom in?
15
I've got a
to
grab.
20.
Thanks.
Could probably push in a bit on that.
This is perfect.
All right, nice. Okay, so the EH meter is actually still going down.
Do you want to hold it there for a
Let's
little
hold there
longer?
for a minute.
Okay.
We're going to take the EH sensor away.
Full wide on Argus.
Stand by.
The microbes of the ocean are kind of mobilizing most of the elements that then are fed up into the higher levels of the food chain. So it's coming
Okay,
into
zoom for me,
the
video.
microbial system at a place like this underneath the surface through fluid fractures of methane-rich stuff, and then that is being processed through the sulfur cycle. Some of those elements, they're being
Okay.
the bacteria are being eaten by some of these snails or worms or clams, and then up the food chain. So all of
Nice.
life is dependent on some of these microbial communities that move our essential elements from
Yeah.
geology and geochemistry into biology.
A little water in there.
I don't know what this stuff in the middle is. His pointy bits.
Yeah, let's take a look.
Yeah,
Woo, sorry, too much.
They look
like
kind
little
of
worms.
like worms.
They do.
That's
They look
Cool.
wormy to me. Yeah,
Are
that's
any of
really
them
cool.
moving?
yeah, they definitely are.
That's probably full zoom, isn't it?
It sure is.
All right. We need microscopic zoom.
An extender.
So Jeff, are these clams? Are these the type that have a symbiotic relationship with the?
Yep. Yeah, that's right. So they host some sulfide oxidizing bacteria in the tissue itself. So that way the microbes are kind of getting a good deal because they're making use of the macro scale feature of the clam and kind of can reach, orders of magnitude farther up than the microbe itself could. So the clam accesses the oxygen in the seawater, and the microbes can mobilize that sulfide in a way that the clam itself could not.
Cool thing about some symbiotic organisms, like the giant tube worms, actually lose all of their internal organs
That's right.
because they don't need them anymore.
Yep. Yeah, as long as the chemicals and the fluids can get in, then they can make their food through the microbes.
You got a big red critter in the
Oh yeah,
top
yeah,
left.
that one.
That's a big one.
Oh, this
What's
is
going on
normal
here?
for a flatfish.
It looks like there's a precipitous
Ooh,
ledge.
Yeah, coming up
boundary.
to, this is cool.
Dramatic.
Yeah, if we can slow down and maybe stop here.
It's definitely the perimeter.
Yep.
Oh, that's
It's got
cool.
probably 30 seconds.
That's a great
And we can
shot.
stop the ship if you want to stay a little
Right.
longer.
Bridge nav, hold position.
Yeah, we get to stop. This is cool.
That's just really cool.
Yeah, let's try to set down and take a closer look at one of these orange covered chimney structures. Yeah, so it really does look, based on the matte distribution, that these little protruding chimney-ish structures are probably higher concentrations of methane-rich fluid. So they aren't, yeah, always on the ground in between them.
How cool is that?
Nice.
There's a sample.
What is it?
It's a pretty big column of stuff.
Steve,
Excellent, Greg.
how long would it take for something like that to accumulate?
That's a really good question.
I don't think they know what it is yet, which makes it very
Yeah.
difficult to
Well, I
determine
know.
how
I
it
mean, even
accumulates.
just rough guesses. I mean, is this 10 years worth of accumulation or 10 million?
I would think it's related to the amount of activity,
Is it kind of like
the
static
amount
mites
of
and that kind of stuff?
seepage.
Yeah, this growth area is rather extensive.
Yeah, nice shot, Argus too.
Yeah, it's a nice shot.
Yeah, and someone's just written in art in the ocean. I think I have seen this in a few modern contemporary art galleries. Oh, we better go for another zoom.
Huge anemones. Actually, those might be Corallimorpharians. The one on the bottom right-hand side, the larger of the three, looks like a Corallimorpharian. Can we zoom in on that middle of that mat? It looks like there's an accumulation
Video,
of
let's push in.
maybe
Clams, right?
mussels or clams.
Yeah.
Yeah, they look exposed though. Yeah.
Hold that.
Look like a long
Must
Oh,
be a-
wow.
clams.
Yep, clams. So you can see the siphons on the clams.
So does that give you an indication as to how old this structure might be?
Not necessarily. I mean, these clams might have a significantly shorter life cycle than
Gotcha. Then we
what could
realize.
possibly precipitate this much carbonate, if it is carbonate.
You want a zoom video?
Of course.
It's a shark.
Yeah, shark.
Oh, nice.
Cool, Shark Week.
Any idea what type?
Probably a file-tail cat shark.
Very prominent gills.
Yeah,
Yeah. Okay.
pretty.
There's a pteropod. I'm seeing a lot of pteropods here.
Okay, go wide again.
Full wide. Thanks.
All right, let's go back over here. Have a
Full
nice
wide on Argus.
current pushing us right to left.
They look like a bunch of minions.
They do.